Hello everyone. This is Journal Club, and today's sociology is presenting about mosquitoes, and besides that, I don't really know much. But yeah, it's your stage, sociology. Take it away. It's your.
Okay, I'm gonna be starting so this paper is on the general process, which is really interesting, but the way that they collected their data on the transgenerational information is really interesting. So this whole process has been theorized before on other papers, and I've looked at little tiny bits. This is the first paper that's able to cover not the entire process, but it's chipping away and it's able to give a good overview of what I'm trying to study. And I think it's a really good paper because of that. And it was done by it's not a new team. This team in Australia just works with malaria mostly. So it's really interesting that they were the first ones to come out with this really interesting paper. So I'll bring up the PowerPoint and show you guys just a few of the figures, and then I can discuss through each of the figures what they're actually going through. Okay,
Oh,
so we're talking about antiviral immunity of RNA viruses. So these are single strand RNA viruses. We'll talk about the
oh,
two different types of them. But these viruses are getting into the mosquito through their blood meals or just whenever they're larvae from the environment. And often they transmit genes or they just want to spread throughout different mosquitoes, but they can also be flaviviruses, which can then get into humans and cause encephalopathy and a lot of other issues. So researching these is very important for both understanding how the mosquitoes deal with them and understanding how we can deal with them while affecting humans. And
oh,
this Dorothy Institute is working mostly with malaria, so it's really interesting that they are the first ones to elucidate this pathway in a really interesting way. So I'm going to start with the first figure. These are just some of the mock infected are ones that they've never seen the virus before, and then the parents were virus infected. That means that their generation before was infected, and so this is how they are transferring immunity. So infected mosquitoes on day zero all have the same amount of virus RNA within their body, but over the days you can see that there's less virus RNA in the second generation, and it's because they have a form of immunity, so they're fighting off that virus. Can you guys see my mouse, or should I use the little pen? You can see first generations here, they're fighting off that virus, and in the second generation, they have resistance to it, and so over a few days, they are reducing down that virus. And so these are just different virus proteins that they're fighting against. So if they have a longer genome, they're able to fight it more effectively, even the initial infected ones. So they're
oh,
less susceptible to SFV infection.
oh,
And so that's the reduction in susceptibility.
oh,
And so this happens within their first blood meal. So they're taking, so how
oh,
they used it was that they infected the blood with SFE, and this is their defense against it between generations. Other ones will do it in their water, or they'll do it in their sugar. But for this experiment, it's within their first blood meal. So these are all female mosquitoes,
oh,
and this is only for alpha viruses. Other viruses will actually stay in the mosquitoes, so it's constantly fighting it. So it'll always have immunity. Will always have some partial immunity to it for those ones.
oh,
Again, these are mock infections, so this is ones that have never been exposed to the virus before, and that's the percentage of the virus itself. And you can see between their first generation and second generation, if they've never been exposed, they have like a high percentage of SFE in the mosquitoes after their first lay and second lay. But for ones that have been infected with different versions of um, so if they've been infected in their previous generation with this FG, so their F1 is the F1 generation, and it's been fed with infected SFE blood before. The challenge is now how much virus is in this next generation, and it has it has 4-star significance lower infectivity of the actual virus. That's what these ones are showing. All the other ones are PCR tests, which aren't looking at the actual percentage of the SFE, which is calculated by calculating how many viral particles there are. So where are they actually being infected within the mosquitoes? That's what this is showing in days post blood meal. So within the whole mosquito, you can see. So these are their infection. So this is the first generation, and you see that over time, over 9 days, it's slowly increasing the amount of SFE as it infects, and it's found in the whole mosquito in its midgut where it first was infected. It's actually going down because it's spreading throughout the body. In the heads, it starts getting infected. It starts infecting there after day 5, and in the carcass, that's the rest of the body without the ovaries, the legs, or the midgut. We actually dissect mosquitoes the same way in our lab, where we're comparing the carcass to the ovaries in our results. But you can see that they don't infect the ovaries, so it's not the virus itself can't go to the next generation for SFE. Now, other viruses that aren't alpha viruses will have infectivity in the ovaries, so they spread between generations. So it makes it harder to be able to research this sort of transgenerational immunity. So that's why they're showing this. So it's consistent with other alpha viruses like chikungunya, which infects humans, and that's spread from their first cycle to their second one. So if they had immunity to chikungunya, we could have less of that virus in the mosquito. So there's some research on trying to keep chikungunya resistance within their genome, but we'll see it actually comes as an issue later on how they're actually stored, and that's what they actually are elucidating. But right now, we see how the virus actually infects the mosquitoes. It's slowly spreading throughout its body, and as it spreads throughout its body, it's not getting into the ovaries for next generation, but it is available within its heads, within its legs. So if it bites another human after being infected with a alpha virus, it can then spread that alpha virus to another human. And that's the concerning part.
Oh,
Flaviviruses can be transmitted vertically. That means it can get into the ovaries or get into the oviduct where it would actually cover the eggs, and then they'll have that flavivirus in them. And that's concerning for other issues. So certain encephalopathy, certain diseases in Aedes mosquitoes will, so they can actually survive in the desert for months at a time. And if their eggs are laid in an area that's very drought prone, you'd expect that there would be no infected animals there because they would be culled after a few generations because there are actually a whole bunch of farming. This is especially in Africa where different farming groups will move their herds around depending on the season, depending on the year. So they might only come to this area to graze every 5 years. But because they're transmitted vertically, whenever there's a flood in that region, these infected mosquitoes will come back and then start infecting the animals again, which is not a good idea because it's not good for the animals because they can cause abortions in the animals spontaneously, and if it infects humans from that, there is a chance for some very serious encephalopathy. So that's swelling in the brain, and that's not very good. This is an issue in Japan and in Africa mostly though. And then so this is this figure is for the larva. So you can see this is the next generation where SFE is not getting in there. RRV, which is another alpha virus, is not getting in there. But dengue can get to the next generation. So that's why flaviviruses were not chosen for this experiment. But the same idea of immunity will is important for understanding how they're fighting these viruses and why it's not as effective on flaviviruses.
oh,
So this is the difference between them. They're just focus on the actual stranding of it. So the positive strand is what's sent over. So the alpha virus is to the left, where you can see it's actually it's turning back into its positive form before it can make its proteins, while in flaviviruses, it's only turning into its positive form for its progeny RNA. So this is already being packaged up, and it only has to make a little bit of that. while for alpha viruses, all of its proteins have to be produced from the RNA form. So all those structural proteins are dependent on one side of that RNA strand. And because of that, it's really easy to find that in the nucleus and be able to eradicate that. So transgenerational immunity is a lot easier to work with, but it's still possible to attack the negative strand with custom gene engineering if we knew how to make it work.
Well,
So how are they collecting? How do they know where it's being spread? Well, they're now able to get
well,
these SFV infected mosquitoes and and this is based off of previous models where they were looking at um, how do we like where are these being transmitted? They theorized that they were coming off of these things called transposons. Transposons will basically take them like they embed themselves in the DNA of the mosquito, but as the mosquito cell divides, or if there's any sort of activity within even within humans, transposons are just basically broken viruses. They don't have any structural proteins, but they do have reverse transcriptase on them, which allows them to be cropped out of the DNA or copied out of the DNA and then be replicated. So these are turned back into either RNA or they're turned into negative strand DNA that can then get back into the genome in other places. But they found that infected mosquitoes are using these copia-like TY1 in TY1 based systems, and they have pieces of SFV inside of them. So this F1, again, let me get the pen out. So these F1 generations of these, after they've been infected with SFV, so these ones haven't been infected, but they had the previous generation. They can they can actually test and find out which ones have pieces of the actual SFE in them. And the way that they extracted these is actually interesting. Um, we we didn't know a good way of extracting it, but it turns out standard kits that you use for taking out um, or taking they're called they're episomes. And so those are, if you know, in bacteria or in yeast, episomes are plasmids. So these are things that people work with in genetic engineering all the time with. And they just took a standard kit for extracting plasmids and they were able to take out all of these and then identify that they were part of the SFE immunity because they contain pieces of SFE in there that they're using to defend against. So they actually use pieces of the RNA of the virus in order, like they collect them in these transposons and then they put them in their genome. And they found out that copia-like and gypsy, so that it's from the spongy moth. It used to be called a gypsy moth. It's an old term for it. But these are from our transposons that should be renamed personally because I don't like using the bad name for them. But piggyback is piggyback is a standard transposon that everyone uses. This high collection of piggyback over here is probably due to it being in a lab with other genetically engineered mosquitoes. Because piggyback is found mostly in moths, just like the copia-like and the ones that are in the spongy moths. But these two are also found on baculoviruses. So copia and spongy moth based transposons are found on the ones that I'm using to modify these. So that's a bit concerning, but that means they have a specific type of transposon system that they're using to collect these pieces and use their reverse transcription to embed it into their own genome again. And they found them by just looking at those little ends of the actual fragments that they were able to collect. So they crop them up and then they're able to find them and they're able to find them in the genome. And that's why they know which types of transposons they are. But it's really interesting to see that the transposon signature changes between a infected mosquito and a mosquito that was just collected and was never infected with SFE. So they're constantly producing these transposons, and they're actually exploiting them as a form of immunity, which is really, really interesting. So there's some sort of system involved in that they're trying to find.
Oh,
Okay. And so AZT, this is something that blocks transposon. So AZT is a protein that we use in human drugs. If you look over here, AZT is just a short name for this drug that we use for people who have certain viral diseases. So someone with AIDS might be treated with AZT in order to reduce viral activity, and it reduces the actual SF. Well, it reduces all transposon activity pretty much, and they're able to see within the challenges that they react the same way. So that at least are reinfecting the second generation with SFE to see if they're still immune to them or not. And then this is the transgenerational check on there. And yeah, so AZT minus, they have these. So this is a qPCR. Well, this is just a PCR really, but they're using it to just measure that pieces of the SFV from these the pieces of SFV from these slides a couple behind. They're actually taking these pieces and then checking them again
oh,
against the genome. So AZT minus, they're able to see it in generation one and generation two, where so that's generation after they've been infected with SFE, and these generations have these pieces of DNA, I mean, these transposons that are resistant to SFE. But when you actually use AZT in their blood meal with SFE, well, actually, no, this is in their sugar ball. So this is actually before they're infected. Their transposon activity is knocked out. The AZT positive shows that they're not able to produce um, these
oh,
episomes, these plasmids that are able to block viral infection. So that's why they're looking at AZT, because it's a system that's actually blocking this whole system that they want to look at. And so AZT minus, SFE on their mock and under AZT minus, they don't have any infected. But this is actually particular.
oh,
So
oh,
this is not looking at
oh,
the SFE
oh, oh, oh,
ones that haven't been infected with SFE at all. So without mock and with AZT minus, so these are just this has not been infected with SFE whatsoever. AZT minus is this challenge right here where it's been given SFV and it has not been given AZT. The next generation doesn't have any virus alpha virus in it, and whenever it's given AZT, it's not able to resist those viruses. And for whatever reason, because it's not able to resist those viruses, it's now being able to be infected transgenerationally. So it's working like flaviviruses again. And so it's constantly fighting this AZT. I mean, it's only 1 out of 5, but that's really interesting because that means that it's blocking all sorts of antiviral defenses that is trying to protect its ovaries. So there is some sort of system that's trying to protect this. Now, it's trying to protect the ovaries from viral infections, and it's failing without transposons. So this is a very essential system for them to resist. So if I'm going to be modifying my mosquitoes to overexpress this system, then I can't use a um, a a baculovirus, because a baculovirus has the same transposons that they're using to block the. I mean, they're using, like, the off-target effect um, would actually suggest that my results are better than what they actually are. And so I need to be very careful about which sort of virus I use and making sure that they don't have any of these transposons attached to them. And that's a major concern whenever I go into actually being able to elucidate the system further, because right now they just have one way of blocking the system from working. So when we get into this major whole system, The parental generation gets infected with SFE. The viral RNA genome is showing up. And in order to defend itself, the mosquito, in order to defend itself from the viral RNA, will reverse transcribe it and integrate it via a transposon into its DNA. And then it will transcribe that via the piRNA system or via just just RNAi in general in order to block this virus from continuing to infect. Even whenever reverse transcription is fully blocked, it'll actually partially block this system from being able to work whatsoever. Even though this dicer system should be able to work directly with the viral RNA without any of this, but we saw that 1 out of 5 were actually unable to use this system by itself to defend against viral RNAs. So this part of the system is what they're blocking with AZT, and it's a very important system for them to be able to transfer it to subsequent generations. So because it's part of their viral DNA as a transposon, it will have RNAi immunity to other SFE infections in the future, where if you block that reverse transcription, it's unable to use that. So we don't know what system is actually detecting this reverse transcription or how it knows to integrate. So what we're looking at is this process right here in our work, and we're using a flavivirus, we're not using a flavivirus, we're using a baculovirus in order to use dicer to actually block some of these proteins that we know are in this system. But because we know that baculovirus-based transposons are a major part of this integration, we have to alter how I'm going to deploy and detect off-target effects, because if I see a change in transposons, it might just be from viral infections. So I have to make my mock infections a little bit better and control for that. But it's really interesting that they're able to fully map out like what this pathway looks like, but not what proteins are involved with this, because that's like the main thing. Because if we can understand this reverse transcription or how it's integrated, we could integrate our own viral immune responses to flaviviruses, to potentially bacterial infections, or to I just lost what they're called, or to stuff like malaria, because we also have a project going on which is looking at the dsRNAs that are being produced through this system, and they're interacting with both the bacteria and phage in there. It's like actually integrating parts of that in there whenever we run it on Kraken. So Kraken's a software that just detects, okay, what's within these, what's within the episomes. And there's some interesting information within these strands, not within the episomes, sorry, within the dsRNA, because we can collect dsRNA really easily because they're always a certain length. But because we know about this system here, we can look at what's actually being collected. And there's pieces of phage, there's pieces of bacteria. There's we don't work with the malaria, but there has been suggestions that they work with viruses that infect those. That work around those. So there's certain proteins within the actual gut. So it's actually doing RNAi against some proteins within the mosquito's own system to be able to knock down transcription. So this system is also used for knocking down other proteins within the mosquito itself. So there's really interesting systems within this RNAi response, but we want to be able to see how they're integrated and see whether we can modify that. So having a paper that actually elucidates this part of the pathway and elucidates ways of being able to block certain parts of it allows us to fully understand this part by being able to modify these parts of the system. Is there any
Oh,
questions? Yeah,
oh,
here's their some of their papers. Some of their like challenge systems I didn't include in here.
oh.
So for the mosquitoes, are they like sort of bats where they have an exceptionally robust immune system that doesn't necessarily like super take a lot of damage from all these weird viruses hanging out inside them?
They have some of the most complex systems that don't have antibodies. It's just either innate immunity or this sort of adaptive immune system where they are like even using parts of viruses to block other viruses, which is really interesting
And
because
this is
these transposons are like normally would kill the mosquito immediately because like baculoviruses are basically the kind of viruses that if you're infected, you are dead within like that generation because it normally turns your whole insides into goo. So ones that can survive with it are somehow able to take parts of the viruses that are the absolute worst and use it against other more simpler viruses. So it's an amazingly adaptive system.
that's, that's fascinating. Thank you.
Now, with bats, I mean, they're taking in blood from multiple animals, so they have to be like resistant to. I mean, it's the same thing with mosquitoes for certain mosquitoes, because some of them will just only prey on birds or only prey on on certain animals. But for Aedes, the Aedes has this most developed system with this endogenous viral RNA system, where it's able to make these episomes with pieces of virus that we know of. So our Anopheles do not have this system, this transgenerational immunity using transposons. We don't see that except only in this species of Aedes or in tiger Aedes too, I guess. So
Do you
that's
think
like
that
the tropical sort of Aedes.
huh, do you think that contributes to Aedes spread around the planet is their ability to handle these weird pathogens?
I think the ability of Aedes to infect multiple types of animals um, is partially to do with that. So it's a chicken and the egg sort of thing. Like, is it because that they're able to bite certain animals? Is that why they have such a high immune system, or is it because they have such a high immune system that allows them to affect other animals and then spread everywhere? Because like Anopheline mosquitoes will are like there's specific species for each one, and they're pretty isolated to one or two species of animals. While for Aedes, they can attack most birds and humans and livestock, and they'll go after pretty much anything. Yeah, so like yellow fever, West Nile, Zika encephalopathy, and equine encephalopathy. So they're these sorts of viruses are able to be addressed with this sort of immune system that they have for transgenerational immunity.
Do you think that's a kind of immunity that can be conferred to a mammalian system?
I think that we have similar systems within our bodies. So, um, whenever we are, so in cancer cases, for instance, you can detect transposon activity related with cancer. And I think as a cellular stress response, we are releasing more transposons. And normally that means that the cell is really stressed, but we also have our own innate antiviral systems as well. So um, mammals don't have such a complex collection system. We have antibodies for that to attack the proteins, but transposons are a part of pretty much mammalian systems, insect systems, like so piggyback is being used in humans because they do, like because it's a transposon that affects humans just like mosquitoes, but it's so alien to us that it actually has very low effect. Different like you can put it into us and it's not part of any previous viruses, so it's not innately being destroyed. It doesn't have that many off-target effects, but for other transposons that we've been dealing with for a long time, like HPV and stuff like that, there's many that are integrated within our cells right now, and they often cause cancer or they're often related with cancer as like cellular stress. Sorry, I went on a weird tangent on there, but I think that these systems are innate to whichever species they're for, because like even mosquitoes don't have, even all mosquitoes don't have the system. But I think transposons and immunity, there's a connection between them for pretty much all animals. So the reason why they're using the SFE M cherry for these M cherry and for GFP, it's actually um, it's actually more related with these parts of it are a lot more easier to access. So this system is dependent on like the length of the actual virus itself. So viruses want to be as small and compact as possible. So if you add a protein like M cherry or EGFP to it, it actually exposes it a little bit more. So they're doing it because it's multiple viral infections, and they were trying to see if it went down, and it kind of goes down a little bit, but it's not really a significant difference. But these increased lengths of DNA give access for not for most RNAi for small hairpin RNAs, but for it's what they're called piRNA systems. So this is still RNAi, but piRNA is actually it binds to it and then it's able to collect more information about it. So there's some interesting methods that they used in here for looking at EGFP. I think they discuss it in their discussion about it. But okay, that's another tangent that's interesting, but I'm not talking about piRNAs today. PiRNAs are just what we're looking at in the end to actually see where these where they're attacking. Other questions we got?
What have you learned that kind of really surprised you about mosquitoes and working with them?
Well, I didn't realize that the baculoviruses were such a major part of mosquitoes and insects in general. So whenever I think about baculoviruses, I thought that they were only related with moths, and because like baculoviruses and moths are a major like so if you think about moths in nature, you walk around and you see caterpillars that are just completely destroyed. Caterpillars have a rough time because they there's billions of them to start with, but over half of them died of just viral infections from plants that they're eating on, and then another half of them of the remaining half get infected by get attacked by like endoparasites. So looking at wasps and stuff like that. But then the rest of them have to grow up super big, and then they're defenseless again, other than a chrysalis for the or a cocoon for actually growing up into an adult moth. And throughout that whole life cycle, they still have major effects on crops and everything. So their research has looked into these baculoviruses since like the 80s or 90s, but we never really compared them with other species, with mosquitoes and stuff like that, on how baculovirus, because they don't infect humans. We just know that they infect moths, and moths, whenever they're invasive species, are really, really bad because there's no viruses that can attack them. So they develop these baculovirus-based pesticides, and that's where all the research was. Like my one of my not my professors, but one of my coworkers, she's not working on a degree program. She has her PhD in moth research, and she moved back from industry on making those pesticides into working here. And so for the past like 30 years, she's been working just with trying to make those pesticides. And it's super interesting on like she didn't like she finds this research really interesting with mosquitoes and these connections with it. So that's partially why I'm interested in using baculoviruses. It's like, oh, we have all this research on baculoviruses and they shouldn't affect mosquitoes in the same way. I then, whenever we actually look at it, the transposons in there are all baculovirus based transposons. Like piggyback in there, okay, that's fine. That's from a specific baculovirus that I'm working with, but we can remove that. but seeing how it's integrating all of these different types of baculovirus, like copia-like ones, or ones from moths that from sphinx moths that shouldn't infect mosquitoes, but they do. It's like, what mosquito is going around and collecting these? It must be a larvae at some point where a moth gets infected, and then the virus spreads in the water for a bit, because those viruses are long lived, but there isn't really a time where a mosquito is on a plant other than when it's a male is collecting sap. So the interaction between those didn't seem like it would be there, but it's kind of like how one sort of infection you wouldn't expect to be between two different species, like say a penguin and a human, like they're in completely separate environments, but then somehow they both can be infected by the flu, like that. That specifically I found really interesting. And then how resilient mosquitoes are. They absolutely need water, and you have to give them water every day, or they immediately dry up and die. So like for like weird small things, they're super weak to. But if you grow them up right, they have amazing fecundity. Like they won't die out on you just with some water and some yeast. Like most mosquitoes can survive like that, and then all you need to do is just give them a drop of blood, and then they're able to make another 100 so mosquitoes, which is kind of insane. Like it's like the scale of things. So like they're only taking a little bit out of you, but they're able to make so much out of that.
What are they actually getting out of the blood in terms of nutrients?
So the blood itself is actually really important for them to be able to get to the next generation. So um, they're taking the proteins out of that, digesting them, and using them to make their albumin and everything for their eggs. So the whole midgut, at least for mosquitoes, changes in morphology, and it's moving all of that stuff into fat, and then those go into fat cells in the mosquito, and then this whole change happens within 48 hours, where like they're redesigning their whole midgut to be able to turn all of those proteins in the blood into something that can then be put into eggs. And it's breaking down anything that they can get from there, but it's looking at specifically the albumin in the blood in order to be able to actually start that whole process.
Oh, oh,
So it has like a hormone, like it's not a hormone, but well, actually, it might be a hormonal response to just detecting this smallest bit of blood, and they change their whole internal structure within 48 hours to start laying.
oh,
That's
oh,
amazing. Um,
I've made some really interesting figures on that because we have to time everything so specifically in order to do that. So for our RNAi injection, so we're actually injecting RNA right now into our mosquitoes, and then we wait. Well, actually, sorry, we blood feed the mosquitoes at exactly 11 a.m., and then we have to come in at 11 a.m. 24 hours later and inject them at a specific stage so it spreads throughout their body at the correct moment before they lay another 24 hours later. Um, and we have to time that specifically in order to get the comparable results with other papers, because if we don't do it at that exact time, it's not going to be able to spread around enough in order to get in to get near the follicle cells. It's never going to be able to get into the actual ovary. We see that in previous papers, but we want to be able to see that in the carcass that we spread it across the body. It's similar to that SFE infection figure in here, where they show it in different regions after different days. Or is that one? Yeah. So we will see it in the legs after 5 days and stuff like that. But after 24 hours, we want it to move. Yeah, we want it to move into this carcass area. So 0 days, it's not going to show up in the carcass, because like whenever we inject, we're injecting it into the thorax, but it needs to move into the into the thorax, but it needs to get to the abdomen within 24 hours. And we realized that 24 hours gets us the best results. Injecting it beforehand, they get they don't actually take up blood, and it's interesting on the timing of these things that you have to do it at the exact hour or it's not going to work. And that's why one of our projects that my major professor was working on before she came here was working on trying to find proteins that are activated at those specific points in time and then use those to then activate a gene that we add in. So we insert a gene into the mosquito that is only activated at that specific point in time, and then we can put whatever we want in that cassette, so we don't have to inject so precisely, and it gets those same consistent results, which is really nice. But we're not using that for our project because we're using RNAi injections, the standard method, to compare against.
Have you guys considered swabbing commonly used sap sources and pollen sources for these male mosquitoes, like looking for baculoviruses on their pollination, I don't know, hosts?
It is exceptionally hard to get male mosquitoes. So
Really?
we can get female mosquitoes in the wild super easily because whenever they want to lay, we can set up an ovi trap. And so basically, normally female mosquitoes are attracted to blood. We can attract the female mosquitoes with blood, and they get the ones that haven't laid yet. Or we can collect the mosquitoes that want to lay. That's what we've been doing because we want to look at what the mosquitoes have been taking up in their blood meals and what in those transposons, what are they actually interacting with in the wild? Because all of these results beforehand have been done in the lab with controlled SFV, but what bacteria are they interacting with that they don't see in the lab because we're feeding them fish food and stuff like that, where we're basically feeding them yeast. That's the kind of fish food that we're feeding them. But like, what are they seeing in the wild? We have a project looking at that, but I don't know how they're able to collect male mosquitoes because all they do is look out for water sources and then collect from there, and then they breed with the female mosquitoes and then they die. So we don't really have a way of attracting those guys other than taking the next generation and looking at what's in there. And there's another thing interesting with the genetics of that. So I don't know if you guys are aware of haploid diploidity. I believe that's what it is. Where it's the same as bee genetics, where males only have half the genome, so they are pretty much only designed to just to spread genes and to mate with other female mosquitoes. So they don't actually have as interesting as a system to look at. Like if you see bumblebee drones, they have half the genes that a bumblebee worker would have, for instance, or a bumblebee queen has the same genes as her sisters, but not not her sisters, as her daughters. But the drone that she made with is just half a genome. It's literally only there to spread that piece. It's basically a flying sperm.
What's the population of like male to female mosquitoes? Is it known?
Males, it depends. So if the female has not mated, so this is with other insects where if that insect hasn't mated, it will release only drones. But sometimes it will aphids will mate with themselves, like they'll mate with themselves within their parents. It's really kind of weird stuff. But in terms of mosquitoes, it's a 50-50 ratio for what we see whenever we take out the first ones to actually come out are the males. They're usually smaller, like the smaller larvae as well. and then they'll become pupae first, and we can pull them out of the water and then put them in the in their in the pupae, like, so we're putting them in their cages where they're going to become adult mosquitoes, and we have to actually make sure that we collect multiple days because this was a mistake that I made before where first day there's a whole bunch of pupae that come out, but they're all males, and if you don't put in another egg cup from the next day, you're only going to have males in that egg cup. So like the ratio changes between cages if you have so many eggs, so you have to split them up correctly in order to have the correct ratio of males to females, because you only need a couple of males in there to for all of them to breed. But it's interesting because they're all just physically smaller. They're because they're all one line. They're all basically clones of each other for what we're looking at in these lines. And you normally don't see wild male mosquitoes because they look like chironomids in the wild.
When you get bit by a mosquito now, do you like think about the poop of viruses that they carry around, or is it just sort of like, oh,
So they there's a whole ton of like if if I was going to have kids in the future, I'd be very concerned with Zika because that's coming around here. But normally, the viruses that are being spread around by mosquitoes are controlled by your local government. So they have these things called sentinel chickens. And these sentinel chickens are detecting whether certain viruses are being spread around. Because if the Aedes mosquitoes bites the chicken will be infected a lot easier than the humans will. It's like a canary, basically, a canary in the coal mine. And Zika is a very concerning thing because encephalopathy is not a fun thing to go through. And then if you or a loved one is planning on having children and you're infected with Zika, then that'd be very concerning. And so I would, if that was a option in the future, or if anything, I would move out before trying to do that because that's the only really concerning thing that I would have with that. So it's like I'm more aware of the sorts of diseases in my area and ones that can spread because if one bites you, you don't know whether that mosquito had it or not, and there's so many disastrous outcomes from that. But as as an adult male, if I get infected with something from mosquito, unless I am living in a malaria region where I'd be vaccinated against it, or at least have be aware of where to get drugs to treatment and for treatment and everything like that, it wouldn't be an issue. But if I was a child or if I was planning on having children, then that would be a concern that I would have in the future.
Oh,
well, thank you so much for answering my questions. I really appreciate it.
oh, oh.
I mean, this is just the overview of what I'm trying to look at, and I found that this paper covered that whole system pretty well for like some of the really interesting parts that 80s like 80s can do. And I mean, I find it really interesting, and it unfortunately this paper has shown that I need to reconstruct some of my methods to cancel out a lot of the likely issues with using a baculovirus. So um, I'm going to have off-target effects that are directly related to some of the results that they're making in this paper, and I need to find out like I'm gonna have to make a whole bunch of negative controls and positive controls if I want to extend this. And it's looks like it's turning a master's project, which was just infecting these and looking at the results, in looking at making a method for it, into making a whole project on elucidating that part of the pathway, which is nice because I would like to go into more computational side of it, and I need data to get that. But on the other side, that means another 2 years of working on this project.
Oh, so when you mentioned how like if you were to have children or if you were a child, like how long does it stay in your system? Like if you get the Zika from a mosquito.
I do not know. I know encephalopathy. So this is like a viral infection that can reach to your brain and it causes swelling within your brain and brainstem, and those cause major nerve system damage later in your life. And as a child, I had a nerve issue in the back of my neck. It wasn't related to encephalopathy, but I had an arthritis issue, and it ended up that I have tinnitus because of it, and I would not. And I assume that being infected with that at such a young age would be very bad for your developing brain, as it would cause some permanent issues depending on how old you are. And then for older people, encephalopathy can be deadly. But for very young children, I think that's like the main concern with these, because mosquito, like the flaviviruses, their main issue with those is not healthy adults. It's more children and elderly, or livestock that are mostly affected by these. And so that's the scary part of them, is it's like those guys, I mean, children are outside a lot more frequently, and in regions where they don't have mosquito netting, if they didn't have if they didn't have access to vaccines or treatments for it, it's a very easy disease to die from, like yellow fever, that causes jaundice. So this is, instead of encephalopathy, it's affecting your liver, and so your liver shuts down as a child, and you start turning yellow, and it's really bad. Um, those sorts of diseases and stuff like that can be life destroying for very young children, and that's the concerning part about these diseases, is that they're not that effective on adults, and so it can spread pretty easily through there. And then the ones that are affected by it are the children. um I mean, that's the same thing with other viruses that we get. Just like coronavirus doesn't affect us, but it affects mostly, well, it does affect us. We get sick for a few days, but it dramatically affects people of different age groups, like the elderly. And their defenses to it are a lot worse, and they respond a lot worse to it. And that's why we have, oh, that's why we try to protect the elderly from those infected diseases earlier. It's the same thing, but for younger people with flaviviruses, because they're outdoors more and they're more susceptible to being bit by mosquitoes.
Oh,
Well, is there any other questions about my work, or my evolving opinions about having to rewrite a lot of stuff and have to replan out stuff over the summer before I can um, work with the actual experiment, because we are working on our baselines right now. We are starting to collect our data this week. So we finished your experiment a couple weeks ago, and we're going to do our first qPCRs on that data now. They're in the freezer, ready to go on Thursday. But it's daunting, because this summer I got to get all this through, and it I might have to. So there's a conference soon that I'm going to be paying $50 to join the actual community for it. But if I present there, I might be able to enter a fellowship. And if I'm able to get funding for a project like this, I might move it into a PhD program, and be able to actually elucidate this whole pathway, which would be very interesting. That requires a lot of biocomputational work, which I am confident in, because I just did a project looking at this entirely computationally. But I want to be able to collect the actual transcriptomes, and that requires a lot more money to do so.
oh, yeah, that sounds cool. Well, I was interested if like I haven't heard any cases of like kids dying from like diseases like yellow fever around here, but is that a
No,
thing that
it's
happens?
not here. We've been able to fight yellow fever pretty easily now.
I see.
But similar diseases like so similar other blood diseases related to with that are hepatitis A. B is like, it has to be between blood transmission, but those caught like they're morphologically similar, I believe. But those sorts of diseases are related with yellow fever, and we've been able to um, to to fight those, but it's still an issue in certain regions. Certainly not as much as what it was in the 1800s now, but now it's these sorts of alpha viruses like Zika that are coming around, which are instead of affecting the liver, which we can treat really well, it's the brainstem, and the brainstem is still has lasting damage if you're a child and you're infected.
I see. Okay. Oh,
Did you
oh,
say something, Jay?
Yeah, I said thank you for presenting. That's awesome.
oh,
Oh, thank you.
oh,
Is
oh,
my volume pretty low?
Yeah,
oh.
I had to boost it, and I was able to hear.
That might be settings. 1 second.
I mean, if we still have time, I'm totally willing to listen to you talking about the summer stuff too. That seems pretty cool. Oh,
Well, that's that. I mean, I think I've presented on that before with I have the PowerPoint for that somewhere actually, because I've updated it for a conference that I did in Toluca a little while ago. I think I presented that to you guys, but that's being moved into proposal. But this new paper, like, I put like, oh, looking for off-target effects as like a slide at the end, but this paper is now, I'm going to have to discuss this paper because it's talking about how transposons that are directly related to what I want to look at, what's being produced from those transposons, is also being
oh,
produced by Bacillus like transposons. It means I'm going to have to look at those using the same process where they are able to use the plasmid extraction kits and look at that. And I don't know how many mosquitoes that requires. So it's going to be contacting new people and adding new methods to the process, which is daunting.
I see.
But our cells are living. We have not gotten the virus to infect the cells, so we take them off cells. We infect them with the virus because the virus doesn't spread in the mosquito. Like the virus that we're using doesn't spread in mosquitoes. We're just using it as a deployment mechanism. That's why I'm hoping that the transposons aren't a major thing, but we have to check, and that requires us dissecting the ovaries before they're laid, and then looking at if the transposon activity changes. But yeah, I mean, if you saw in the previous one, there's not. I mean, the main thing that mosquito needs to do is protect their ovaries from being infected with a virus, because then that means that the next generation's gonna not be. It's gonna have a lower fecundity, and so evolutionarily they have like even in humans. I mean, our human ovaries are trying to avoid any sort of viral infection whatsoever, because that would mess up the next generation's DNA. And um, like we've accumulated a lot of viruses over time, but um, that is one of the most conserved systems that needs to be there. The, the actual eggs themselves needs to be able to be the same between each generation. So trying to modify and look at that is so complicated. Um, I really don't want to lose the ability to infect those follicle cells, and only baculovirus seems to be able to get into the follicle cells, which we'll see in our dissections. But I'm the process that I found to get in there using developmental biology that I want to test to get into the actual eggs. If that gets any baculovirus in there, then it ruins the project. So it's really concerning that if I see anything in there within the mock infections that we do over the summer, that my project won't work out. So it's scary, but I mean I can still write a paper around that.
Oh, I see. I see. Okay.
I think I presented about that before with the baculovirus. Yeah, I did. Um, the I I don't know if the recording's up anywhere, but basically I'm using the baculovirus not to infect the eggs themselves to modify the system, but the follicle cells which surround the egg, and then using a maternal RNA, I'm able to have it be moved into the actual oocyte itself, and then because of how the actual egg is developed, there's a large cytoskeleton in there which will move it to one side of the mosquito, the posterior region, and then that's where I can perform the RNAi without using so I'm not using a transposon anywhere in there. But if the baculovirus itself is changing the transposon signature, that's the concern. But because of how I'm moving it in there, the baculovirus itself isn't infective. It's just deploying a piece of DNA that will turn to RNA that then has the activity that I want it to do. There's likely off-target effects. I just don't want the off-target effects to be directly related to what I want to look at. So it just means I have to do an additional stage before I can actually work with my cassette.
I see. Oh yeah. How at least like with you like collecting the mosquitoes, has like the climate crisis affected that in any way? I know like it has around different areas, but like are
So
you
where we're collecting those mosquitoes does change.
Mhm.
So I'm not the one that's working on the project of actually collecting those mosquitoes. That's what Evan's project's working on, and they are working on their project's actually pretty great, but they're collecting the mosquitoes in our local region, and then they're able to they have to move out to other regions because most of the lakes in our area are already artificial, but there's enough natural 80s around there that we can collect a lot of data about that. But because our I mean our winters have been longer, so this summer is like probably the best chance that we have of collecting mosquitoes because they don't really live that well in the winter, while biting flies will do very well in the winter. So that's partially an issue where like we're the mosquitoes that we're collecting aren't the 80s mosquitoes anymore. They're the Tiger 80s, which are the ones that spread Zika, or they're the or we have these other chironomids that are getting in there, or flies. So it's actually negatively impacting the number of mosquitoes that we're getting, which is weird because like there used to be mosquitoes everywhere here, but there's actually less of them here now. And it might be just due to the way our water system's now working, where these because like within the past 30 years, Lake Dallas is actually filled up where it wasn't filled up a long time ago. And so these are new systems where mosquitoes first got in, but now that the water system has been changed and stuff like that, and the water is now has fish in there and everything, I think that these ecosystems are still like establishing themselves. And as new animals come in, it seems like the number of mosquitoes has actually decreased, which is kind of concerning for what we want. But we just moved to different regions and we're able to collect mosquitoes.
I see. Okay. Wrap up time?
Yeah, I guess it's time. I could talk about Tanvi's project and how she's working with like she just finished her master's thesis on this, where she was working with mosquitoes that have been grown sterilely, so like we can have like a negative control to look at. But there was issues with that project, so it's still ongoing over the summer as well, where basically we grow up the mosquitoes in a sterile water condition, and then we grow and in antibiotic condition, and then we feed them antibiotic laced sugar balls and blood, and then we go to the next generation, and those ones should be sterile eggs, and we want to be able to look at what sort of transposons are still active. Just as a background, so we're running like transcriptomics on that, but we weren't able to collect those because we weren't able to collect enough mosquitoes from the first one, just enough to write a paper on it. But in the future, hopefully over the summer, I'll be able to actually collect those mosquitoes and get a background to compare with our data of the mosquitoes that we're collecting in the wild. You also have mosquitoes that have glowing brains that we just collected recently, which is really interesting. So these mosquitoes with glowing brains, we're using for dissection training. So we had someone from I just lost what her name is, but she's from the Czech Republic that works in a lab in Irvine who brought over 80s mosquitoes that we are using for training for looking at other forms of dissection. So like those head dissections aren't the only thing that we can do. We can extract the salivary glands and see what's in there, or we can extract the brains and see what's actually in there and see whether baculovirus can infect those regions. But those sorts of micro surgeries to collect those tissues are super intense. So we're training on those. So that's why those glowing brain mosquitoes we got from them just to train on them, which I find really interesting because they found a way to modify a mosquito that looks absolutely normal, but if you cut them open, they have EGFP in their brains, so you'll be able to see it only in neural tissue.
I see. Okay.
Yeah, but that's not really. I don't know what project we're actually going to be using that for because I don't think I need to look at brain tissue, but it's interesting to be able to look at the systems. And we're growing up that line, which I hope that we can actually, because that's another thing that Evan's been working on is actually learning how to dissect those, and they've been doing a great job on that. But Tavi and her project, that's the only other one that we're working majorly on the summer. Collecting those mosquitoes is going to be, it's mainly a passive project where we're collecting the eggs from them, and then we can use the eggs that are collected from those OV traps to then grow up and then sequence.
I see.
Because a mosquito, whenever they get into the OV trap, they lay and then they die. So we look at the next generation, but because we're looking at Aedes mosquitoes, once you dry out those eggs, we can look at them at any time in the future. So it's just passive collection right now, while we work on growing up mosquitoes for these big headline projects.
I see.
I think I've said everything that we're working on this summer. So, thanks for showing up and listening. That's it. Thank you.
Yeah,
If you have
thank you
any
so
other
much,
questions, you
Sadoji.
can DM me.
Oh, yeah,
My DMs are
asphalt
open.
philosopher. I saw you like on mic, but I was wondering.
You might be asphalt. You might have to use the push to talk button because we're not hearing anything.
Or you can type out your question. Either way. I guess that wraps up today's journal club. Thank y'all so much for coming. Thank you so much, Sazoji, for presenting. This was a
Thank you
wonderful
for showing up
presentation.
on a weird day. I'm sorry that I wasn't able to do it tomorrow.
Oh no, it's no worries. It's totally fine. This was really cool though. Yeah.
Yeah, I just wanted to give a status update on the project. It's mostly me having a desperate cry for a crisis in faith on my project, but I think the research that they did here was really interesting, and from a team that's new just like us. So, well, not new for this type of research, which I found really interesting. So
I see.
their methods are cool. I want to replicate what they did.
Well, thank y'all everyone for coming. If anyone wants to present in Journal Club, y'all just like shoot me or Gabe a DM, or just in the Journal Club chat. And otherwise, I guess have a great night or day or lunch, I guess.